Thank you very much. Yeah, so just to give a quick overview of what I intend to cover. So I'll firstly just give a kind of quick summary of how clinical drug development goes ahead, what the phases of that are. Then I'll give an overview of the Agile platform trial, which I was involved in design of. And then I'll focus on the design for evaluating the efficacy within that overall trial, because that's what I had the most involvement in. And then at the end, I'll sort of give a quick summary of what the kind of real world outcomes or impact of the research was. Okay, so when people think about clinical trials, probably what you're most likely to be thinking about is actually sort of the phase 3 clinical trial. So that's like the large scale confirmatory trial where you've typically got a head-to-head between the new treatment and a control or placebo. But before that stage, a drug needs to go through a series of other stages. So phase 1 is dealing very much with the safety and the tolerability of the particular drug. And then phase 2 is doing things like demonstrating whether or not there's some evidence or promise of efficacy, and also deciding what's the best dose and dosing schedule. And it was really this phase 1 and phase 2A was what I was involved in this Agile trial. And then the other bit of background is that this Agile trial that I was involved in is a platform trial. So again, traditionally, when a pharmaceutical company is developing a drug, they would have set up their own bespoke trial or trials for those different phases. But recently, there started to be a trend towards what are known as master protocol trials. So this is the idea of allowing either multiple treatments or possibly evaluating multiple different conditions within the same overarching design or trial infrastructure. And a special case of this is the platform trial, where specifically we're looking at the case where we're looking at patients of the same type, so patients with, in this case, COVID-19, but we want to evaluate multiple treatments. So potentially not just one drug, multiple candidate drugs for that particular condition. And these platform trials have various benefits. So on the one hand, they sort of guarantee or make it much more likely that we're able to get sufficient recruitment of patients. Particularly if one's already running a company, would know that they're going to be able to get the patients. It should reduce the overall costs of evaluating a treatment. And what was most pertinent in our situation was it can speed up how quickly we can get a result for a particular evaluation of a particular compound. So these platform trials really came to the fore in the spring of 2020 with the emergence of COVID-19, because people wanted to try and find effective treatments as quickly as possible. So there was a phase 3 platform trial that was set up, the recovery platform, that was looking at repurposing existing treatments to see whether they would also have efficacy for COVID-19. But the one I was involved in was this Agile, which is a phase 1/2 platform. So it has these dual aims of looking to establish whether the drug is safe and which doses are safe for that candidate drug. And then, if that's established, to then gain an indication of whether we also have efficacy for those safe doses. And so here's a kind of quick diagrammatic representation of the trial design. So the main idea is that patients would be recruited into the trial in batches. And so they'd be randomized to some will just receive a placebo, some will receive active treatment. When it's the first patients on a new dose treatment combination, then the first couple of batches of patients will be assessed for safety. So that's looking at the rate of dose limiting responses, so sort of toxicity events. And so after those first couple of batches, there's a first decision made as to whether to proceed with that particular dose because it's deemed to be safe, or to terminate due to safety concerns. If we deem it to be safe, then more batches are recruited for that particular dose level. And then we evaluate for efficacy using a Bayesian survival model. And here we have a series of interims. After each batch has had 28 days of follow-up, we're going to decide whether to continue evaluating, and we can decide to stop early, either because we think the results so far are sufficiently promising that we've got enough evidence to say it can proceed to phase 3, or there's not enough evidence, and so we're stopping for futility. And the idea is that you can have multiple batches of these patients, potentially on different doses or at different treatments at the same time, and that's going to further help us with the design. In terms of how to evaluate this efficacy, we've basically got a time to event outcome. So specifically, efficacy endpoint was the time to a 2-point improvement based on the WHO clinical patient scale within a 28-day period. So the idea is they're evaluated each day, and if they've improved by at least 2 points by a particular day, that's their endpoint. They get to 28 days without improving, then they'll be censored at 28 days. If they die within the 28-day period, that would also be treated as effectively their improvement time is infinite. So it's equivalent in this case as taking it as 28 days effectively. It's a time to event endpoint. So the standard approach to this is a Cox proportional hazards model, which is saying that if we've kind of got 2 patients at a particular point in time, let's say 15 days, and neither of them have had this improvement yet, then it's saying that the effect of treatment would be, for any particular time point you considered like that, the instantaneous risk, or you can think of it as a sort of probability of having the improvement on that particular day, will be effectively sort of 75% higher for the treatment arm than the treatment patient and the placebo, if it's an effective treatment. So this panel on the left is basically giving us a hypothetical of what we might expect to see in terms of the data we would collect if we were looking at an effective treatment. And the good thing about this approach is, although it's making assumptions about how treatment affects the time to event, it doesn't make any assumptions about the shape of that time to improvement distribution. So the idea of the design specifically is we'd have a maximum of 12 batches of patients, each involving 4 patients on active treatment, so the experimental, and 2 on control or placebo. But we're also exploiting the fact that we'd have other recent patients who would have been evaluated for other dose levels or possibly other different treatments, who were given the control, which we can include in the data we use to evaluate how much better or worse the new treatment is compared to control. That's going to help with the efficiency of the design. And so after each batch, we've got the option to make this decision as to whether to proceed or not. And the design has a kind of hybrid of using, so there's kind of 2 branches of statistics. There's sort of the Bayesian paradigm, the frequentist or classical paradigm. This design is kind of using both approaches to some extent, because the decision is based on whether we're above or below thresholds based on the Bayesian posterior probability of efficacy. So it's sort of saying, given all the information we received so far, what's our estimate of the probability that the treatment's effective? If it's higher than some value, we're going to stop for efficacy. If it's lower than some value, we're going to stop for futility, and otherwise we're going to keep going unless we've reached that final batch. But to calibrate what those limits U and L should be, we're going to use classical methods. So we're going to ensure that we control type 1 error. So this is the probability of incorrectly concluding that an ineffective treatment is effective. We're going to constrain that to 10%. And we're also going to try to ensure that the power, so this is the probability that if the treatment really is effective, we are able to conclude that result. We want that to be at least 80%. And so the real sort of computational challenge of devising the design of this efficacy part was in finding the values of this L and this U. And so how I actually went about doing that was basically through Monte Carlo evaluation, so simulating a large number of potential trials under these two different scenarios. So under the scenario where actually the treatment or this particular dose has no effect on time to improvement, and the case where the dose treatment combination is considered to be effective, and you've got this 75% increase in the rate of improvement. And so what we're looking to do is to vary these boundaries to get the right type 1 error and power, and to also try to minimize the expected number of patients we need to get a result. So to start with, we've got a situation where we've got very high upper boundary, very low lower boundary, and we're kind of getting too lower type 1 error, too lower power. So the first thing we could do is look to, well, let's lower the upper limit. And kind of if we do that, we can get to somewhere where we've got much better power, and the type 1 error is kind of going towards the 10% that we want to allow. But we're kind of getting too many people that are, or too many trials. So each one of these kind of trajectories is a different hypothetical trial that might have occurred. We're getting too many cases where, although the treatment's ineffective, we're going and using all of the possible batches of patients. So what we can then do is say, well, if we instead also look to increase this lower bound, then we can kind of get to the sweet spot where we've got the right power, we've got the right type 1 error, and we're kind of minimizing the expected number of patients that we need. So the overall design is kind of saying that after each of these batches, apart from the first one, which we just assume we don't make a decision after the first batch because 2 batches are needed for safety anyway, we would make the decision based on this posterior probability. If it's greater than 84%, we'd stop now for efficacy. If it's less than 22%, we'd stop now for utility. And by doing that approach, we only need about 30 patients on average. And this is very favorable compared to what we might need if we use the kind of conventional non-adaptive design, where we just have no early stopping, no use of past controls. For that, you'd need to use all 72 available patients to achieve the same power and type 1 error. A further aspect of this, so obviously what I did there is just, that's kind of for one dose treatment combination. We also did further simulations to consider how the overall design would work with respect to evaluating multiple doses. So specifically, we considered that there might be 3 candidate doses and different scenarios for safety and efficacy of the different doses. And then what we found is sort of the worst case scenario where all doses are safe, but none of them work. The type 1 error, so the overall chance of recommending an ineffective treatment as being effective is around 12%. And then the kind of power depends heavily on the kind of, how many of the doses are safe and how many of them, and how steep the kind of dose response curve is. In the best case scenario, you might be able to get an overall power of 82%. It's much worse if in difficult situations where maybe only one dose is considered safe or effective. But the overall conclusion was that by using this design, you were going to rarely recommend either unsafe or ineffective treatments or dose levels. And then, so finally, what then happened in practice? So I was only really involved in the design of this trial. So the work I did was in the spring of 2020. It got approved at that time. I think the first patient started being recruited in July of 2020. And then various treatments have been evaluated in this agile platform. Most notably, Merck first trialed their drug, molnupiravir, and it had promising results, and then eventually graduated to phase 3 confirmatory trials. And this was the first oral antiviral for COVID-19 that was approved by the UK's MHRA in November 2021. And then fast on the heels of that, another drug that was evaluated within Agile by Glaxo, this sotrovimab, was approved in December 2021. Each of these are kind of for higher risk patients who've got moderate to mild to moderate COVID-19. So yeah, finally, here's just some links or references related to the research. And I should certainly acknowledge this is very much joint work with Professor Thomas Yaki, Dr. Pavel Mozganov, who both of whom had much more of an involvement in the practical implementation of the platform, and also joint work with Dr. Helen Burnett. Okay.